I moved from Melbourne back to the city because tired of country living. I want to be together with people. And I look into the report from City of College Station. So they do have the same measure for the same group of chemicals that in that similar range of the reporting, the city water doesn't have this issue. Okay. So what I bring this to your attention is how can we see what our eyes cannot see? And the philosophy or the principle is very similar to the pandemic, like COVID. And in the early time of the pandemic, some studies suggest the rate of asymptomatic infection could be as high as 80%. Then the latest study revealed approximately 17% of the population, they have a presentation of asymptomatic, even though they probably have the COVID virus in their body. What we can really find out about those asymptomatic patients is only through testing. And many of you know that in the early days of the pandemic, that because there is not enough tests available, then there was public outcry crisis as well. So this brings back to a numeric interpretation of Ali's introduction for me. And I want to use some numbers to show you our operation when I served as the division director back in Iowa. So our division has 3 sites across the state, and on an annual basis, we conduct more than 155,000 tests in water, and we have about 75 FTE in our operation. And out of those tests we do in our lab, more than 100,000 are conducted in water, then about more than 20,000 was conducted in private well. And we have a lot of advanced instrumentation to support our testing. Now I will bring this to you as a case study, then also present to you a research project we're conducting right now. And what I want to use is an element, or it's a metal, arsenic. I'm pretty sure many of you are not strangers to this element, and you know it has potential health risks to human body. And then I will present to you of a public health and environmental health strategy by using surveillance sampling and testing to understand what's the health implication of this element in the general population. So EPA regards any form of arsenic to be toxic. And back in 2001, CDC regards arsenic is the #1 in hazardous substances in the Superfund sites. And then the International Agency for Research on Cancer regards arsenic to be a group 1 or group A carcinogen. And many of you probably have watched the news, read those articles on internet, and then get those research updates from different feeds. So the image here on the left shows you a CNN report of discovery of arsenic in apple juice. And then FDA says their tests showed no toxicity. And it's not surprising, occasionally or often you will hear rice is very easy to contaminate with arsenic. Then on the image here on the right shows you a study from University of Washington. They found that arsenic in many US red wines and actually potential in many other drinks, but their study suggests health risks depend on the total diet. I teach a class, the title is Water and Bioenvironmental Sciences. So I keep teaching that class and emphasize to my students saying that the particular chemical could be toxic, pathogens in a similar way, but what most important is dosage and exposure length. You can eat apple, and apple is healthy, but if you eat a ton of apple, that can be toxic to you because your health will not be able to upstand that tolerance level. So what I want to emphasize is arsenic is truly a toxic chemical and a carcinogen. Then depending on what you are used, particularly in drinking water, and where do you live, then your risk of exposure to this toxic element is different. This was a study conducted by USGS, United States Geological Survey, as a collaboration with CDC. So this is a global national estimate of the population that can be exposed to arsenic that's higher than 10 microgram per liter, which is the EPA maximum contamination level setting limits for drinking water. So you can see different location and different population size have different exposure risks to arsenic. Then the picture here on the right shows you a Texas study that has been conducted by a group in University of Texas in 2018 that shows you different color and then the potential arsenic exposure risks. So again, I cannot say the audience and how your response to this map, but you can map of your current location or maybe where you have moved. Then you can potentially estimate the risk of arsenic presence in groundwater. So globally, this is a paper published in Science in 2020. This is a modeling study to estimate what could be the potential of arsenic exposure and presence in groundwater. So, but again, you can see the whole world is not evenly distributed upon those studies. We have more data in developed countries and like in North America, like in Europe, and part of the Asia, part of the Africa, but not all over the world, evenly distributed. But you can see, again, depending where you live and depending on what kind of water source you use for your drinking water, people's exposure to the risk can be very different. So as I mentioned, what our eyes cannot see, but you can get the information is through a testing. So I'm showing you an image from the TCEQ, Texas Commission on Environmental Quality. They do have a list of the qualified labs you can find, and then you can take a sample, and then you can do a measurement. So this brings me to this program we call Private Well Program that we established back in Iowa, and it's also named as grants to counties. So in that state, the state of Iowa, the program is coming from the state funding, then the Department of Public Health, as well as the Department of Natural Resources, which is equivalent of the TCEQ in Texas. Then they managed together a program that the state funding has been allocated to each county, that if the county works with a local citizen who has a private well, so they can work with a household and to do a free test. And then if there is an issue, then they can come up with supporting fund, not enough to repair the whole well, but as a supplement, so the household can do something to make sure that drinking water is good. As part of the program, then our lab has supported a lot of arsenic testing in the state. So this is a spatial distribution of the arsenic testing we have performed in Iowa since 2015. Again, you can say uneven distribution of testing across the states. In this image, you can see two different colors. If the dot is in red color, then it suggests that well has been tested, contains arsenic, exceed the EPA Safe Drinking Water Standard, MCL, more than 10 ppb. If that color is blue for the dot, then presumably that well at the time of testing contains arsenic less than the 10 ppb, which is a safe drinking water standard. So what we want to do in this particular case, we have done a lot of testing, but we know we never have enough resource to test all of the wells. So we looked into the correlation of the test. And the picture here shows you on the left is we hope to understand if we can estimate the arsenic contamination risk in different locations. And then many of the times people will think, well, you test so you know, and if you test more, then of course, potentially you can say more risk in that region. But I'm showing you those two picture image comparison to say that in our study, the risk study identification is not because the more tests we do, it's a real correlation between the sampling and the location. So the picture here on the left shows you the number of tested wells in each county in Iowa. Then there are 3 counties show a gray color that we don't have any data in those 3 counties. Then the image on the right shows you the sample proportion of contaminated wells among all the observed tests for each county in Iowa. And you can clearly say the pattern of those observations are not the same, which suggests it's not because you do the test that you have more risk of detecting that arsenic contamination in that location. So I will bring this very briefly, discuss this concept very briefly of what's the rationale behind this particular study in this project. So I will mention for the probability theory and the binomial distribution. And then we use this to estimate, we use this theory to estimate the probability of arsenic present in the private well that is higher than the EPA MCL, basically the safe drinking water standards, 10 microgram per liter. Then we assume if we regard the presence of arsenic in that private well is more than 10 or less than 10 to be binomial distribution. If it's more than 10, we say it's a yes. If it's less than 10, we say it's a no. Then by doing this treatment, we can presumably assume that the presence of arsenic higher or less than 10 potentially can follow a binomial distribution in that state. Then because of this distribution, we can estimate what could be the percentage of the arsenic risk in that particular location. So one thing is underlying in this assumption is the sample distribution of a count variable is only well described by the binomial distribution in case the population size is significantly larger than the sample size. So what's the population size? And Iowa does have a database constructed from 2000. So this map shows you how many wells have records in that database. So our total population in this case is more than or close to 300,000. So our assumption is pretty safe here that given the large population of more than 300,000, then what our sample size will be significantly smaller than that. So while we're approaching this phenomenon observation with this question, and what I am regarding as important is really from a management perspective. And in living these modern days, we always know there are more problems, there are more information, and problems are more, issues are more, but money is less. So it's hard to really find the resource, identify the resource to fix those issues. So efficient sampling in this case would be very important. So you can efficiently allocate your limited resource toward those management in terms of large population, in terms of public health and environmental health. So we came up with this spatially adaptive sampling design that will estimate a spatially clustered underlying contamination distribution. So basically, it's different from those traditional spatial sampling design that often assume there's a continuous process-based spatial mode. Then the model underlying the contamination risk is a spatially clustered function. So basically, we assume if you can detect arsenic in those locations, then spatially, you may be able to see a cluster around that area. So that's one of the underlying assumptions in our study. Then the advantage of taking this modeling is we can detect this continuous spatial heterogeneity in the arsenic distribution, which we know a lot because arsenic could be present in this location, but you may not have it. We know it could be as a local issue, but still it's a lot of heterogeneity in that distribution. Then we can borrow the information within each identified spatially homogeneous cluster from an adaptive sampling design. So by applying that model to the population or to the data we have collected and analyzed, I showed you the map of the spatial distribution in the first present slides when I began to talk about this story. Then we identified that 3 risk clusters in the state based on the data we have. The dark black color shows you the lowest risk. Then from our analysis, we regard the risk for this region is low. Then the magenta color or the purple color shows you the cluster 2, which means the potential of the people living in this area, then the groundwater or the private well they use to get their drinking water is higher than the people who is living in the cluster 1. Then this is not new to Iowa because we have done a lot of study, and then we know certain counties in that region has a potential high risk of arsenic contamination. But through this study, we identified a new hotspot, which is a yellow color on the border with Nebraska. We found out that cluster 3 actually present an even higher risk compared to those conventionally or traditionally understood observation of arsenic contamination in the central region of Iowa. So based on this analysis, we came up with a future sampling saying that we need to sample more samples from cluster 1 because we know the sampling has not been taken enough data to further validate the sampling study. Then in cluster 2, there are other regions we still want to further do the study. So the blue color shows you the potential new sampling location. Then of course in cluster 3, that's one of the priorities. We hope we can get more sample from that region. So this is part of the historical analysis of the data we have in our database. So what we did as another outreach or research project is starting from 2017, when I was back in Iowa, we started this program called Iowa Well Survey. So in that study, we outreach to those counties. As we know in the past, we have certain collaboration on certain parameters that we test for water quality. But how about the emerging contaminants? And those are not even in the EPA regulation. People don't know the health risk. People don't even heard about the presence of those things could be in their water. One example is neonicotinoids. I don't know how many of you heard of this from the news. This is a newer generation of pesticide, can be potentially very harmful to the bee population. And there are confirmed studies to show the neonicotinoid can be very harmful to certain bee population. Some studies think it's not generally applicable to all the bees, but again, science has not been finalized the conclusion. So I use this as an example that we outreach to the counties. So we work with them, then we collect a sample, then we take the additional sample for those emerging contamination study, and we do this study twice a year. Starting from fall 2017, I showed you the maps. We worked in the past with different counties from 2018, 2019, and then to currently. The program was unfortunately partially paused due to the pandemic because out of those human interaction restrictions during the pandemic. So this brings as a foundation for a program we are conducting right now, which has been supported or sponsored by CDC from 2019. I continue to serve as the director for that program. And I don't want to say thank you to COVID, but again, because of the COVID, a lot of virtual working make things possible. So what's biomonitoring? Biomonitoring is a study that in analytical chemistry, we can measure the body burden of a toxic chemical compounds, elements, or their metabolites in biological substances. Then you can see the chemical can exist in many environmental matrices. It can exist in air, in soil, in water, in products, in our food, and other things. If you think of the potential exposure, those things can get into our body. We can eat them, we can drink them, we can touch them, then those chemicals will stay in our body depending on the property of the chemical and depending on the person's handling capacity, that a chemical can potentially stay in our body for a long time. So what we propose to CDC and has also been sponsored by CDC is we work with 10 counties, then we work with the population at risk who is relying on private well for their drinking water. Then in this study, we analyze heavy metal, we analyze pesticide, as I mentioned, neonicotinoid, one of the newer generation of the pesticide, and then environmental phenols such as BPA, BPF, and other chemicals has been used in plastics and has been included in a lot of personal hygiene products. Then we want to take a sample from the private well. Then we're also going to solicit a biospecimen, in this case is a urine sample. Then we can make a correlation between the drinking water exposure and the potential chemical residue in the human body. And another part of this study is we also assess the maternal exposure to metals and pesticides during pregnancy. So we're using a residual sample from the tissue bank as a collaboration with University of Iowa Hospital and clinics. So this is an ongoing project, as I mentioned to you. It started in 2019, and in the past 2 years, we spent a lot of time to launch the program, planning our recruitment theme. So we're at a stage that our institutional review board has approved our recruitment protocol. We're proceeding with recruiting the real people into our program. And I hope we can stay tuned so I can come back and talk to you for the progress on this exciting project we are performing right now. So as Arlie also introduced, part of my program also include environmental remediation approach. As I mentioned to you, if we have a population, if we do the test, we can identify the potential risk. But what ultimately would be very important is what can we do to mitigate and manage those risks? Of course, to reduce the human exposure, and then a remediation for what has been contaminated is very, very important. Population, sampling, testing, and remediation is what I think a treatment train for that, how we deal with a lot of those environmental issues together with water quality. So again, I have my web page, and I don't know if I happen to include that in the flyer for the presentation. But what my lab on the website, wet lab side, we have a lot of interest in doing those chemical assessment, and we try to understand the molecular mechanism and characterization for those bioremediation. So we use bacteria, fungus, those microbes to come up with ways to detoxify and degrade those environmental persistent chemicals. And we also do a lot of data. So I showed you some examples of the data analytics we do so we can understand and estimate those environmental risks in water. One example is PFAS. I don't know if you heard of these things in the news or reading articles in the past or not. PFAS includes a group of chemicals, more than 6,000, has been synthesized by human beings since the late 60s. And then the structure of those PFAS I have presented here in the middle of this slide. You can see the carbon is connected with fluorine, and those compounds contains a lot of fluorine, replaced hydrogen. And those compounds are so beautifully designed. They have very attractive properties with different applications in the industry, in medical treatment, and in the food packaging industry in the past. But again, because of the very attractive nature, and also contributes to some of the very undesired property, they're very, very stable. So as if they're released into the environment, they can stay there forever. And I mean, saying forever compared to the lifespan of a human being. Then EPA has worked with different stakeholders beginning in 2004 or 2006. And again, EPA has continuously issued different updates on the potential perspective of regulation. And it's going to be regulated by EPA, I'm pretty sure, from a personal interpretation. But right now, it's not in the drinking water standards yet by national level. But there are many other states have already established their state PFAS, like compliance levels in water. And then I apply different platforms to study those, depending on the goal of my research project. So one of the platforms I want to introduce to you is I use this so-called shotgun proteomics. And then it's a wonderful tool that you can identify a lot of proteins, thousands of the proteins in the samples you want to analyze. It can be a plant, it can be a soil, it can be a fungus, it can be a bacteria, it can be a human tissue. Again, depending on what kind of the stuff you want to analyze, as long as you can extract protein efficiently from that sample, then potentially you can use this tool to help your research goal. Then I want to give you some idea of the protein dynamics, and then the dynamic range and a challenge in regarding to the protein analysis from the real sample. When we talk about those housekeeping soluble proteins, they're present in the specimen or in those biological samples at 1 to 10 picomolar concentration. But if you talk about soluble proteins, then they're less concentrated. Come to membrane protein and those PTM, post-translation modification proteins, then they're present at even less concentration, which creates a huge challenge in those analysis if you look into the whole proteome of the species or the samples they are looking at. Then mass spectrometry is one of the most advanced techniques we can use for those proteomics analysis. If you have a well-established platform and with good quality control, then you can reach FMO sequence sensitivity in those protein detects. So this is also a cartoon presentation of the platform I use in my lab. So we do a so-called like two-dimensional separation of the peptides when we get the protein extracted. So we digest the sample into small peptides, then we separate by two-dimensional chromatography, then we use mass spec and analyze those peptides, and then we do a data search. Go back to the database that has been constructed by a target organism, fungus, bacteria, human body, plants, as long as you can think. Then we do a database search by doing the informatics, then we identify what are the proteins identified in this whole global proteome analysis. So this is one example of the raw data and then how we do the data analysis. Now I want to give you another example of like a wet lab, how we understand and find out tools to understand those presence of the chemical and how we can come up with an effective way to manage those contaminants in water. So in this example is an organic dye, and we are using fungus, white rod fungus, to say how effectively this microbial can degrade the organic dye in water. And what we discovered is white rod fungus has been well known for being able to degrade plant walls. And if you buy a corn, you put it in your refrigerator, in 2 weeks, you're going to see mold. Those fungus are very effective degrader for those plant tissues, especially white rod fungus are very well-known eaters for the cell wall from the plants. Because of this very unique nature, people discovered white rod fungus can degrade a lot of environmental contaminants present in the environment. Then thinking of the white rod fungus can live on the plant cell, which are the natural substance. And then what it prompted us to look into is what if we use the plant cell residue, like lignin, as a component or substrate, and we mix that together with a chemical we want to remediate as a contaminant as a pollutant in the water, for example. Then in this case, we did discover a very interesting phenomenon that if we mix the lignin, which is a natural substrate for fungus, together with the organic dye, then the degradation efficiency for dye was significantly improved. Then we did an analysis of the proteomics, try to understand what are the important enzyme groups in this particular phenomenon. And we did discover, very interestingly, a huge group of redox, which are related to reduction and oxidation process of the proteins in the whole process. A lot of interesting enzymes has been either upregulated or downregulated, depending on their job in doing the degradation, together with dye and lignin. So promoted by this idea, which make me to think, if lignin is something that's wonderful for the microbial, it can promote the degradation of the dye. What if I use lignin as a natural substrate and we build something that the fungus can eat, but at the same time, this stuff can use as an absorbent to enrich those chemicals in the environment, then we can kill, you can kill the bird with one stone, right? So what we come up is this so-called nanostructured artificial plant material. So use lignin and the components from the plant cell, then we build this porous material, which can be used to treat water. And then after the water is treated, we can retain the chemical from water, then we give it back to the fungus to say if the fungus can utilize this as a food as well as degrade the chemical at the same time. So in this case, not only dye, we are moving into PFAS. So the picture here on the left shows you the composite we built by doing the microscope analysis. And then we have used this new NAP, so-called nanostructured artificial plant, to treat PFAS. And we found efficient retain capacity of PFAS on this material. Then we grow fungus and compare without using this material as a substrate. Then the fungus cannot even survive when the water is containing 10 ppb of the PFAS. However, if we use this material, then the fungus can grow pretty well on the material that has been treated with very high concentration of PFAS. On the picture here on the right shows you we have treated this plant material with more than 10 milligram per mil of the PFAS, and then the fungus can still grow on that material. However, if it's liquid media, then the fungus cannot even survive at 10 nanogram per milliliter. Then we also tested the absorbance capacity of this material with PFAS, and we compare with different composition when identify the most ideal artificial plant that can be used both for chemical treatment and then also as food for fungus to grow. So on the bottom right here shows you that the image of how the fungus is growing on that NAP substrate. So what's happening behind the thing? The picture here shows you on the left, shows our comparison of different composites we have built in our experiment. And the picture here on the left in labeled with A, showing there's no growth of fungus in the control, then very better grow on one of the composites we developed. And then the figure D shows you it's our control, which grows fungus, but not optimal. And then we also further with this process characterization with high resolution mass spec. And because we want to understand what has been the degraded product from PFAS, then this is one of the identifications of the metabolites from the fungus degradation with our potential proposed pathway. So if the PFAS is longer chain with carbon molecules, then through those fungus degradation, it can get into shorter by removing one carbon and another carbon, and potentially, I hope, we'll still continue doing the project right now with the hope that we can reduce this to a much even shorter chain to reduce the toxicity of PFAS. So one of the things I don't like Zoom presentation is I cannot see the interaction from the audience. And I want to give you this question is, from your estimate, what do you think in your mind? How much does the US household spend on water? Again, I cannot see your face and cannot see if you have any answer to that. I want to present this image to you so that's the past estimate, but I'm pretty sure this will stay stable as for future years to come. So on average, the US household spend close to 50% of our utility bills on phone, internet, and cables. We spend about 10% on gas for heat, for cook, and for those purposes. We spend around 90% on electricity, again, for of course, heating and also all those domestic usage. We only spend 4% on water. This refers to drinking water. And we spend another 4% on wastewater and stormwater. So again, I don't have any comments or position on those numbers, but I want to bring those numbers to your attention so we can think. Again, in this society, we pay for what we get, and we invest this into our water facility infrastructure. Then what do we expect for the future to happen is supported what our investment today. So with that, I want to acknowledge a group of people when I work in the State Hygienic Lab, and I'm continuing to work with the team right now to perform that biomonitoring project. And also I have collaborators with A&M, potentially in the future with a prior review and A&M University. that we do this data modeling or bioremediation project. Then currently I have 2 students and then a postdoc working for me, and with funding from CDC, NIH, and as well as A&M. So I will stop share so I can really see my colleagues' face or interaction. And I'm happy to answer the question if you have any.
Well, thank you, Dr. Dai, for this wonderful presentation, very thorough presentation. We'll give you a virtual round of applause. So, and the floor is open for questions and comments from the audience, so please feel free.
Dr. Dai, very interesting presentation. Thank you so much. And what are the plant species you use for the bioremediation of arsenic?
Are you asking for the plant?
Yeah, bioremediation, do you use any plant species?
Yeah, so in this case, actually, we use lignin. It can come in from any plant, because you can even buy that from Sigma. And of course, you can process that from corn stover. It's a very abundant lignin source, but you can get that from woody species. Lignin is universally present in many, many plants.
Yeah, you know, the some plant species naturally absorb inorganic arsenic.
Yes, so very good question. So that's one part of the bioremediation. People use plants and then they grow plants on top of those, like contaminate the soil field. Another thing with that approach is what are going to deal with the biomass, because you can grow those plants on the arsenic contaminated field, then you're going to have a harvest of the biomass, right? That's another, I don't call that a shortcoming. It's another thing you have to deal with, another downstream. I don't call that a waste. It could be a waste in this regard. But again, bioremediation, phytoremediation is a large field that EPA even sponsored a lot of study. And I know in A&M, like many of the colleagues here are using plants to do those bioremediation.
Yeah, and some of our preliminary studies shows we are growing hemp now. Hemp, cannabis absorbs a lot of heavy metals and arsenic from the soil. And also, what is your idea about using poultry manure in agriculture? Because the poultry manure is one of the major source of arsenic contamination.
Yeah, very good.
Any idea?
So manure, we have not worked on manure in this regard, and I know manure has been one of the source like for bioenergy, like the biogas production as well. Very interesting idea. Yes, I think manure can be a potential another waste source if we can process some material from manure, and then we can come up with some good things. And then that's one of the things I remember. We did a project on this type of the work, and the people come to look at the shitty job you are doing.
Yeah, the poultry feed use small amount of arsenic, and so usually poultry manure get a lot of arsenic accumulated. So there's a big question about use of poultry manure in especially the green leaf production and all that. Yeah.
Yeah, very good comments. Yeah, I need to think more about that perspective, because a lot of those like sludge usage, those manure application in the field can be another contamination source if those are not properly treated.
Correct. Thank you.
I think along the same lines, ferns are known, used for bioremediation for arsenic. And I think, Dr. Dai, maybe you're familiar with the work of Dr. Lina Ma at University of Florida. She's done lots of work in this area on using of fern plants as a phytoremediation to remediate arsenic. I think it's Going through arsenic, we published a book on arsenic in water resources with Springer. So I think I'll give you the link for that one. But I want to mention something about your study in Iowa. Have you looked at the geological formation contribution to those areas? Because arsenic can be either from geological formation that is part of the bedrock, but inherited concentration, or from industrial or agricultural operations. So I think knowing the source of the arsenic would help to in any mitigation process or any thing that we need to address the water quality issue. Have you done any work on that area?
Very, very good comment, because I 100%, actually 200% agree with you that arsenic largely depend on the location, the geological configuration, and then those water interaction with those rocks, bedrock, and then a potential newer manipulation of the ground, right? So you're 300% right, because one of the reasons when I discovered that is based on some initial pilot study, like in Sarasota County. And then at that time, there were more studies done because people have not known that issue in the state to understand the aquifer and then the oxidative status or those parameters in water to understand why it has a high concentration in the groundwater. So right now, because of the new data analysis, I present to you for the risk identification in different regions. And that yellow spot is new. So I didn't present to you the potential overlap of that small part with the geological identification or characterization by the geological survey. So that yellow spot is actually part of the Missouri alluvial plain, different from the other part, just align that border of our identification. So right now, I'm trying to get more data to understand what could be on the other side of our border. But sometimes it's not really, if you want, then have the data, because historically, people may not have collect any data in that region. So there's no way for you to know. That's why I'm saying what our eyes cannot say. As researchers, as everybody, same as me in this room, we know that. We have tools, we can identify the risks by doing the study, by doing a test, then by doing analysis. Again, I agree with you 300%. What can be the issue in Texas, right? I show you the map of the groundwater. But again, what's happening in their kitchen? Because there is a large gap in those outreach, and many people even are aware they are probably living in a region that has high arsenic risk. That's why I regard the extension in A&M, extension in prior view in A&M are very important, because you guys are the front runners to disseminate the studies we do in an academia environment.
Yeah, I think that's the same thing with air quality, with radon, for example. Some area have, geologically, they have radon emissions than others. So looking at those background concentration is very important. So I think we want some more questions and comments from the audience, and we welcome those. This is an important topic. It's about air quality and water quality and what we drink and what we eat.
Well, just food for thought. You know, we're in the process of since we're talking about manure, we're in the process of revamping our animal waste handling system out here. So there's an opportunity to sample from the collection cisterns where the waste will collect, also from the settling tank, which is fed by gravity flow from the cisterns on both sides, and then the clarified pond after the material settles out in the clarifying tank. So in terms of the small ruminant and feeds that we use here in the center, there's a number of different ways we could utilize that waste, and there's 3 different collection points. So I think it would be a interesting project to try to utilize that waste in some way, whether it be compost for adding onto the fields or the gardens, and what may be contained in it and how it influences the soil profiles.
Interesting. Yeah. Great.
I think I'll tell you, for example, when I was in Florida, there was a common practice to treat cattle. Is basically they bathe them in an arsenic solution. This is back like decades ago, but those pits have been source for arsenic contamination there. And I think it's Iowa as a state who, I mean, raise cattle, I'm sure, and have corn and soybean, etc. Maybe those pits or those areas that have been used historically are sources of contamination too.
That's very possible. You are correctly pointing out, not only the first question, geological, those are the things you cannot choose. I mean, you can choose to where you want to live, but as long as you buy your house, you're not going to sell that in 2 months. And then environmental change upon the water quality is another thing. So that's why we in the biomonitoring project, we have designed a survey. So we try to capture those information as much as possible, like how far away you live away from a feedlot or some other things.
All right, I think I would like to maybe, since we don't have some additional comments, or like to maybe explore the opportunity that there are potential for collaboration. As Gary mentioned, we have opportunity for waste processing, waste treatment. Also, since we have, I mean, a large water group, water team that are interested in different aspects of water quality, both microbial and also chemical and biochemical. So if there is any opportunity for potential joint projects or potential collaboration, we will be happy to really entertain those type of opportunities. So just keep that in mind. So maybe if there are opportunities, yeah, we welcome those.
Yeah, exactly. I appreciate the time and talking to you guys. Again, I hate we cannot meet in person, and I'm a person truly believe that in-person interactions are crucially important. So I hope everybody get our vaccine, then we can meet very soon.
Well, thank you, everybody. I think we would like to conclude here. So, Dr. Dai, if you have anything to add or closing comments, I would like to thank you. But if you have if you want to make any additional comments before we close here.
I want to thank you for all your time. 50 minutes on Zoom is not a present.
Well, thank you again. I appreciate it. And everybody, thank you for your participation. We appreciate your participation. Give us, our speaker, another round of applause, and you have a wonderful rest of the week.
Thank you. Bye-bye.
Thank you. Bye-bye.
Bye-bye.